So everybody ready, I hope, right? All right, with that, let me just ask Cher to introduce the speaker.
So hi, good morning, everyone. So this is a story. On the wall of my office hangs a stack of photo frames. Inside each frame is a quote, a principle that I live by as a scientist. And one of these quotes reads, "Take care of your people." Dan said that to me many years ago when I was still a struggling postdoc. But today, the limelight is on Dan. And this is in itself an ironic statement, because as you might have guessed already, he would prefer that his students take center stage. And you could just prove this by looking at the track record of his students, many of whom have gone on to become famous authorities in their own fields at prestigious places like the Johns Hopkins, the University of Rochester, University of Minnesota, Oregon Health Sciences, and the Allen Brain Institute, just to name a few. And I think that Dan's top-notch mentoring skills came from his own training with world-renowned scientists. He was a PhD student of Thorsten Wiesel at Harvard. This is the same Wiesel as the Hubble-Wiesel partnership, who won the Nobel Prize in 1981. And together at Rockefeller, they published a highly influential paper about horizontal connections in V1 that has over 1,000 citations to date. This paper sparked a long-term interest into the functional architecture of the visual cortex. And what I mean is this: how circuits are organized in your brain and in mine to enable vision. This endeavor led to over 20 publications with very difficult experiments that saw multiple sleepless nights just to collect neuron data. But of course, what you really want to do is to see those circuits in action. So Dan teamed up with Amiram Greenwald. And if you consider Hubble and Wiesel the gods of visual neuroscience, let's just say that Greenwald is like a demigod. He and Greenwald developed a technique called intrinsic optical imaging that you will hear about today. And actually also on their team is Ron Frostig, who is right here in UCIS faculty in the bioscience department. Their first paper was published in 1990 using signals from blood supplying oxygen to the brain. And remember, this paper came out 2 years before the groundbreaking discovery of functional MRI, something to a similar concept, but using a much more complicated setup. Dan would later go on to reinvent and patent intrinsic optical imaging for the retina. Between the cortex and the retina, he has over 40 original articles to date and over 3,000 citations. To add to this list of contributions, he has served on study sections at the NIH, NSF, as well as being an editor for the Vision Research Journal. His lab has been extensively funded by diverse agencies like Bright Focus, Glaucoma Research, American Epilepsy Society, NIH, NSF, as well as the Navy and DoD. Following the footsteps of his illustrious mentors, he has won several awards, including the prestigious McKnight Scholars and the Whitaker Biomedical Engineering Awards. But you could argue that Dan's contribution is far greater than his credits. This morning, we are very fortunate to have Dan present his quiet journey into giving us intrinsic optical imaging that has become a mainstay for many labs around the world. But even after playing with the gods, he would still humbly turn around and say, "Take care of your people first."
Okay, can everybody hear me?
Yes.
Thank you, Cher, for the overly kind introduction. Hopefully I can measure up to what Cher has previewed to you. And indeed, I thank you again for the invitation to share with you. What I would like to tell you about is a new method, relatively new method for doing functional imaging in the retina in vivo that is economical and yet is non-invasive and can tell us something about the physiology of the retina, and hopefully will have some clinical applications. So, but before I go too much further, as Cher mentioned, I just wanted to um, um, memorialized and notes the huge contributions of Miriam Grünwald to this entire body of work, both in the brain and in the retina. He was tremendously influential in the development of optical methods for studying the nervous system, and we owe a great debt, both myself and, as Cher mentioned, Ron's Frostig, who is also on your faculty. Of course, he passed away recently, but we remember him with every little image that we take of the nervous system. So, if you don't mind, indulge me a little bit in some historical musings from a personal standpoint. My own beginnings of this journey, I can trace back to my undergraduate years, where as a teenager, I started dabbling in things like electronics. In high school, I learned about computers. And in the beginnings of my undergraduate years, I began to learn about the brain. And as I was thinking, I distinctly remember this time, I was thinking about, okay, the brain's a pretty cool organ. Some people like it to a computer, but how can we really understand how it works? And of course, I was familiar with the notion of doing electrophysiology, sticking an electrode, recording neuroactivity, but I, concluded my, undergraduate naive thinking, concluded that you can't go all the way with electrophysiology. There's no way you can stick an electrode in every single neuron in the brain to understand what's going on. So then I thought, well, wouldn't it just be great if neurons just lit up every time they're active? And indeed, that thought, that goal, has driven much of Miriam's and other colleagues' efforts over the past few decades. And this whole line of work starts with that goal, that promise that maybe using optics, wouldn't it just be great if the nervous system would light up very specifically when it's active? And so Miriam started, in fact, there was a small cadre of people in the late 70s that began to investigate optical methods for looking at neural activity. And since we generally thought of the nervous system activity as being electrical, their thought turned to the notion of, could we develop so-called voltage-sensitive dyes that you extrinsically put onto the nervous tissue and cause it to change the property of light, either absorption or fluorescence. And so here's an example of a dye that was created by one of Miriam's chemists. And the idea was to you apply this dye, it gets absorbed across the membrane of neurons, and then you can use an optical setup here, a 10 by 10 diode array, and a computer to monitor the activity of nervous tissue. Here, a cell in a dish. And you see one of the remarkable properties of this that already clearly goes beyond the electrical methods is that you can record from places in the cell that are not just the cell soma. You can record optically in the various processes of the cell and see their response, which often is significantly different than what's available at the soma. Okay, the story quickly moves on to the cortex. I became very interested in visual cortex, started working with Torsten Biesel, recording from visual cortex. And there they had, of course, spent many years charting out the so-called functional organization of the cortex, such as ocular dominance columns, a tremendous discovery of orientation columns and orientation cells, but it was still difficult at what now people call the mesoscale to see the organization of these circuits and cells. I remember very distinctly in 1986, a SFN, Society for Neuroscience meeting, where this researcher, Gary Blaisdell, who had been working with David Hubel, dramatically announced to the world this ability of using voltage-sensitive dyes to visualize very precisely that actual organization of functional cortex. Here you see some examples of his groundbreaking paper of the ocular dominance columns in V1 and orientation columns using voltage-sensitive dyes. And so I remember Miriam's reaction at that meeting, and he was like flabbergasted, and he was going to talk immediately after Gary, and he was basically stunned, but then eventually came to talk about things where his strengths lie. And then later he would look at this data in this paper, and I think I've already told you enough to see there's something not quite right about it. Here's the trace of the optical signal from Gary's work. And you see it's small, 0.1%, and it's very slow on the order of seconds. Whereas I had showed you previously that one of the hallmarks of voltage-sensitive dyes is that it can follow action potentials, that it's very fast. And the signal is very large. The signal here is 4%. So there's something that doesn't quite smell right with this data. And Miriam would later realize that this is not a voltage-sensitive dye signal at all. In fact, Gary admitted that you put the dye on, this blue dye, you put it on, you sit it there, hope the neurons absorb it, but then you wash it off and, well, the brain looks great. Well, if the brain looks great, that means the dye hasn't been inserted into the membrane. And so, as it turns out, what we were looking at was not a voltage-sensitive dye signal at all. And what we would later discover is this is a hemodynamic signal. So then came our work without using any voltage-sensitive dyes at all, in many senses, going and reproducing and providing the same kinds of results that Gary had and extending it beyond in the cortex. And so this, as Cher mentioned, has become a standard method in the field for looking at functional organization in cortex. And here all we're doing is just taking pictures of the brain and looking at the tiny little changes in absorption of light that are due to changes in hemoglobin absorption. What I tell lay people is, supposing I told a dirty joke and it embarrassed you a little bit and you blushed. Well, I can see that your face blushes, that there's more blood going to your face. And if I took a picture of your face, I would capture the state in which more blood was in your face. So that's basically what we're doing. The brain neural tissue blushes. It recruits more hemoglobin when it's active. Okay, so we would take this technique quite far and continue with it today, studying the visual system in other parts of the brain. It's been used in the auditory cortex, somatosensory, motor, and so on. But now I want to tell you about a variant, because as I moved my lab to a new location, I had some downtime, and a friend of mine then at MIT, he had just gotten his PhD with McGunkus and became a glaucoma ophthalmologist as well. And he said, well, you know, I know about your technique. Wouldn't it be great if you could use it on the retina? And so, you know, the idea, which I took him up on it since we had some downtime in between labs, is just to point the camera into the eye instead of onto the surface of the brain. Now, it's a little bit more involved in that, so I modified a standard Topcon fundus camera and created two light paths, one in which the imaging is done in the infrared, because we don't want to, in the cortical experiments, we had imaged in the red part of the spectrum, because there's a big difference between venous blood and arterial blood in the red part of the spectrum. You know that anecdotally yourself. But we don't want to do that in the retina, because that would be blasting the retina with a lot of red light, which would interfere with its visual transduction properties. So what we do is we're imaging in the infrared, where there still is a difference between venous and arterial blood, and we're superimposing via a dichroic mirror a visible stimulus, usually in the green, but it can be in other wavelengths. So there's an infrared imaging path illuminating the retina, and then a visible stimulus path that is stimulating the retina. And here you see in this little movie here, I hope if you can see the movie, which is roughly in real time, the response of the retina in the infrared to a visible vertical green bar. And you see it is on the order of hundreds of milliseconds to a few seconds to develop the full signal, but it's quite spatially specific and only occurs that there's only recruitment of new blood flow where the stimulus is stimulating the retina. We were first done in cats. You see the optic disc here, for example. Anyways, so this is the actual setup, the stereotaxic for the animal, the CCD camera, a Topcon fundus camera that's been modified for these two optical paths. And back then we were in part driven by the notion that this might be good for glaucoma. Counter flickering checkerboards are a classic stimulus for that application. We're always thinking, ganglion cells with center surround fields would respond best to checkerboards. So we devised this stimulus set of both horizontal and vertical bars moving across the field of retina, about 30 to 40 degrees of retina, as well as a full field counter flickering checkerboard. And this, of course, is in the visible part of the spectrum. We collect the data with a block design, a few frames of acquisition without a stimulus, followed by a stimulus, and followed by the time post-stimulus that we record. And so these are the kinds of results that we get. It's notice the signal is very small, as Gary noted, 0.1%, or about 1 part in 1,000. And so, the human visual system isn't good to contrast levels of 0.1%. So if you took a look with the naked eye of these images, you would barely see any difference. You would not be able to detect any difference. But through subtracting the baseline image from the stimulated image of the retina, you get the difference image, which when magnified can reveal the small reflectance change due to the increase in hemoglobin in the stimulated part of the retina. In cats, and I'll try to skirt around this question because it's still a little bit mystifying, but in cats, we see actually two signals. The darkening, which we attribute the most to the hemoglobin absorption, and then side by side, asymmetrically, there is often a white or positive signal, but only in cats that we've seen. We have not seen this in primates or in other animals yet. We think that it's a form of signal stealing. Those involved in fMRI have, people speculate happens in fMRI, so-called blood stealing effect. So it may be that it's going on. But if you trace out these time courses, seconds along here, we often express the signal strength as fractional reflectance change, or delta R over R. And these indeed are on the order of 1 part in 1,000, sometimes 1 part in 100. You see the negative increase, that is a darkening of the retina due to the stimulation, as well as side by side, at least in the cat, often a positive signal. So that's the temporal time course of this signal. It's highly spatially specific. As you see, moving a horizontal bar across different positions in the retina invoke a corresponding darkening that's spatially specific in the retina, such that you can even write your name on the retina. I've written various words on the retina. It is highly spatially specific. Okay. Some properties, point spread function of this signal seems to be on the order of 1 to 200 microns. So pretty good, although not at, single cell level. It increases in strength with increasing stimulus duration. And here I wanted to give a shout out to another one of the key people in this work. I believe you've met with him before. I gathered he gave a talk to your group somewhat earlier. This is Jesse Shalek, who did his PhD work in my lab, working on this project. And he did a number of the seminal experiments trying to figure out these. He's now tenured faculty at the University of Rochester, working on some of these issues using adaptive optics. But he did a lot of the work trying to figure out the answer to these 2 fundamental questions about what I've told you so far, which is, what are the biophysical mechanisms underlying these signals? And then what are the anatomical origins of these signals? And so, I've told you it's hemoglobin, but that was sort of giving you a prelude. How do we know for sure that it's hemoglobin, that it's hemodynamics? And so one set of experiments that Jesse did was using blood contrast agents. The first one, nigrosin, which is basically India ink. It's just something really dark. And you inject it in systemically into the bloodstream, so that if you made a measurement of retinal reflectance and then made an injection of nigrosin afterwards, the retinal reflectance would dramatically drop. Basically, the entire circulatory system gets a lot darker. And now you do this paradigm of optical imaging, and you see there is an increase in the signal size. But so this blue is the before, the magenta is the after, and the green is taking the before signal and scaling it to be the same as the after. And you see what the message is. We have not changed the nature of the signal by injecting nigrosin. We've only changed the magnitude. And so that's a good hint that the signal originates from the circulatory compartment. The action spectra of nigrosin is here. There's a spectrum from 700 to 900 nanometers near infrared. So you take this and you apply it to the intrinsic signal data, and you see that the intrinsic signal data of the imaging spectrally has now shifted. This is the different spectra, has shifted to that which mirrors the absorption curve of nigrosin, which further confirms that it's in the circulatory compartment, and the only thing that's changed is consistent with nigrosin being in the circulatory compartment, increasing the size of this signal. All right, but how about something that's very different? Because nigrosin has a property that's somewhat similar to hemoglobin itself. So we then chose a dye whose spectral properties is very different from nigrosin, indocyanine green, which of course is very commonly used in ophthalmology, has a peak around 800 nanometers. And we did basically the same thing. And you look at the shift in the action spectra of the signal, and it mirrors that more or less of indocyanine green itself. Again, demonstrating we're looking at a circulatory compartment signal. And so this helped establish the hemodynamic nature of our signal. This means that this signal is, of course, very close to what people study with fMRI. And you see Jesse's attempt here to make parallels between the bold signal seen in fMRI and the retinal functional intrinsic signal. So that if you compare published bold signal data, time courses, and so forth, there's a great deal of similarity between the intrinsic signals seen in the retina and bold fMRI. In fact, these two methods have been used to explore details of each other. Optical imaging has been used to further understand the origins of the bold response. And Amiram's been key in that investigation as well. Okay, I think this is the final piece that talks about hemodynamics using nifedipine. You'd predict using a vasodilator might potentially bottom out the vasodilation. So what we're talking about is a vasodilation that is triggered by neural activity, increasing the amount of blood. And so nifedipine is a classic calcium channel vasodilator. And so this is another one of Jesse's experiments. This is mean arterial pressure, and then you give them nifedipine, and sure enough, the pressure is dropping due to vasodilation. And now you track here in the red the behavior of our imaging signal, and when you give them nifedipine, it also drops the intrinsic signal as well, which means that the idea is correct that if our signal depends upon vasodilation and you're already bottom out the vasodilation of the vasculature, then the ability to further vasodilate and see our signal is greatly reduced. So that's all in all the story with nifedipine and confirms the hemodynamic nature of our signal. So the combination of these three classes of experiments leads us to the conclusion, indeed, we are looking at a hemodynamic signal, vasodilation. Okay, the second question is what drives the signal? What drives this vasodilation? And as I recounted to you, our initial thought is that perhaps it would be useful for glaucoma. And so the prediction then is you give, I'm sure you're aware that ganglion cells are the major spiking neurons in the retina. So if you give a agent like TTX that is going to suppress the action potentials, the spiking activity of the ganglion cells, you would expect that our signal would disappear if it's in fact driven by the ganglion cells. Well, the long and short of it is that does not seem to be the case. So we did, thanks to Laura Frischman, we adopted her protocol for doing intravitreal TTX injections. And here's the before, here's the after, and you see it's pretty much the same. So unfortunately, it's unlikely this technique will be that helpful for glaucoma, but we'll get more to that whole question later. So yeah, here's the positive signal. It also remains even with TTX. How do we know the TTX is effective? Two types of control experiments, one using PERG pattern ERG recordings. We see there's a deficit in the ganglion cell contribution of the PERG recordings after the TTX. So we know the TTX is having an effect on the ganglion cell layer, and yet we still have our imaging signal. The other control is the consensual pupillary light reflex. And here again, we confirmed that with TTX, there was no consensual pupillary light reflex, and yet again the optical imaging of the retinal signal still remained. All right, well, if it's not the ganglion cell layer, we got to move up the inner retinal layers and cell types. So the next kind of similar experiment, again, aided by Laura Frischman's advice, was the use of APB and PDA, these glutamate agonists, basically mimic darkness. Remember, the retina is, or the photoreceptors are kind of backwards. They release tremendous amounts of glutamate in the dark. And so these drugs essentially block the on and off pathways by mimicking darkness. And so we made these injections of intravitreal APB and PDA. And well, again, the signal still remains. Before PDA, APB and PDA, afterwards, the signal is still there. So wow, this signal really hangs on and is apparently not dependent on the inner retina at all. We're now talking about blocking the photoreceptor bipolar synapse, and we're still getting this retinal imaging signal. So the inner retina does not seem to be, by and large, involved in the driving of this signal. Well, some further experiments to get at the nature of this signal. Here we see that there's no spatial frequency tuning. So our attempts at using the checkerboard, counterflickering checkerboard, turns out wasn't necessary, that a luminance, mean luminance bar of the same luminance would elicit the same response. And if you have a background of the same mean luminance as your counterflickering checkerboard, you will get no response, basically, because there's no contrast here. Well, actually, shouldn't say that. There's no difference in luminance here. So this is strictly a luminance-based signal, which, well, jumping ahead, is exactly what you'd expect if the signal arises from the outer retina and is driven by photoreceptor activity. There's no center surround response here. Okay, the last nail in the coffin for glaucoma with this method, we managed with the help of Gillian McClellan, a optho vet now at the University of Wisconsin. She sent us two cats with congenital glaucoma. And here you see their IOP measurements are very high. The OCT was done, showing a reduced retinal nerve fiber layer in these cats. Here you can see PERG measurements on these cats, where basically it's for the right eye, the PERG measurement is in line with an optic nerve crush. So basically, very little ganglion cell function left. So it's confirmed that these cats have full-blown glaucoma and very dysfunctional ganglion cell output. And yet we are able to see, again, the imaging signal. So yeah, not going to be good for glaucoma. Everything's pointing to the outer retina as the driving source for these signals. Okay, some more experiments done by Jesse using photo bleach. So you blast the retina with a strong light. Indeed, it does wipe out the signal. Here, this is before the bleach. After the bleach, the signal gets wiped out, and it takes about 20 minutes for it to begin to return. All right. Now, if you look at the classical Wald et al. Bleaching data, you see the cones, they should return their function within about 5 minutes or so. It's only the rods that take something like 20 minutes or more to restore their function after bleaching. So this already suggests to us that we're not looking at cone function here, we're looking at rods, or at least that it's dominated by rods. And of course, rods dominate our retina to begin with. Perhaps not so surprising. Also, they use the most energy of just about any cell in the body. So it's not surprising they would have a very active, well, I'll use the word now, neurovascular coupling mechanism. That is the connection between neural activity and recruitment of blood resources. So here's further bleaching studies done combined with ERG, showing that it takes 20 minutes for this signal to return. And now spectral bleaching, where we just bleached in the blue as well as in the red. And of course, the blue is going to hit the rods very hard, whereas the reds have basically, oh, I guess I should have emphasized that here. So red bleaching is going to have very little effect on the rods, whereas blue bleaching, of course, is going to bleach the rods. And that is what you see here, consistent, again, with the notion that we're seeing a rod-driven signal here. The red bleaching has barely any effect on our imaging signal, whereas the blue bleaching, it again takes about 20 minutes for the signal to return. Okay. So the upshot of these experiments is that now we're looking at a hemodynamic signal that is a negative reflectance signal that's small in amplitude with a rise time on the order of hundreds of milliseconds to seconds. The amplitude is about 0.1 to 1%. It's driven by outer retinal activity, no spatial frequency tuning. It's almost certainly comes from the rods based on action spectra and recovery from bleaching. And we are looking at a hemodynamic process. So that's what we know so far about the signal. Our next step into this story involves rodents. We now switch to the mouse, in part because of the rich genetics available in the mouse world. So I developed this imager for the mouse. It just basically, I don't know if you can see this in the camera, just takes one of these otoscopes, which is meant to be stuck in your ear, and conveniently has both an imaging path as well as a light delivery path. And so that's what we use. It's based on an article in IOVS on how to do this, just to do retinal imaging in rodents, but now we use it to do functional imaging in rodents. So here it is in the mouse, non-stimulated, stimulated. You see that it does, the signal does seem to be present in the mouse retina. The darkening of the retina after the stimulus. The stimulus was so bright it sort of blocked out, it sort of overcame the dichroic mirror basically here. All right. In the mouse, the signal is even smaller, at least in our hands. And so it's a tiny signal, but it's nevertheless there. It's also a negative reflected signal. The retina is getting darker to stimulation. Here shows some spatial specificity. We haven't gotten to be as good as the primate or the cat, but if you take a off-center LED and just move it around, you can move the signal around the mouse retina. Other than the size, so here's the mouse, here's the cat signal. If you scale these two, normalize them. So other than the amplitude, the time course seems pretty similar to the cat signal. All right, what do we do with this mouse? Well, to sort of really tie up the question of where the signal comes from, we got the transducin knockout strain, GNAT, from Janice Lem. And as you probably know, GNAT has a non-functional rod transducin. And so here it's missing the rod response in GNAT. And here's the results from GNAT as opposed to wild type. Well, here's two examples from GNAT showing absolutely flat line, no stimulated response in GNAT. And here, of course, is wild type mouse, GNAT. So GNAT mice having a non-functional rod transducing does not have this signal. Finally, so just think back about what I've been telling you for the past 15 minutes. Everything we tried to do to knock out this signal has failed. That is, it survives TTX, it survives APP, PDA. Bleaching, I guess, does knock it out. Here we see that with the knockout of rod function, rod transduction, the signal does also go away, and confirms that we probably are looking at a rod-dominated signal. Okay. By the way, it also rules out then, because we were speculating, could this be coming from the IPRGCs, melanopsin cells? This suggests that by and large, the answer is going to be no. Okay, very quickly, this just shows that we're able to get signals in the primate, which of course is of great interest because they're going to be closest to us humans. So here is a signal from the macaque retina. It seems to be somewhat smaller than the cat. Maybe this is due to the luminous efficiency of the cat compared to primates, cats being about 2 to 6 times better at capturing light than primates. Here we replicated the TTX experiment in the macaque monkey, showing that before and after TTX, the signal stayed the same. And this is one of my favorite experiments I like to show, using now an LED array in the macaque. We see a high degree of spatial specificity to a point where you can even see individual retinal vessels get turned on in a very tight locale within the retina based on just one of these LEDs being stimulating. So this bodes well for applying this to the human, which of course is one of our ultimate goals or holy grails. So what about the human? Well, unfortunately, humans aren't as easy subjects as animals. They fidget, they complain, they blink. And so it does work, but we're still working out the kinks as to how to better improve the protocol for these humans that don't cooperate nearly as well as a sleeping animal. But you can see the result in the human to a spot, to a vertical bar, and here to a horizontal bar. So this is one of our things that we're still working on quite intently. Okay, I mentioned that all these agents introduced intravitreally do not seem to suppress the signal, so that all the inner retina, the rest of the neural retina does not seem to be involved. And so I remind you here of the APB PDA result. You know, it's hard to get rid of this signal, is it? Well, okay, so what's going on? If it doesn't involve the inner retina, we think it's coming from rods, phototransduction is occurring, hemodynamics is the result. It's not the inner retina that's the connection to the hemodynamics. What is it? Well, the general rubric for this kind of interaction is called neurovascular coupling. And in this case, we're going to invoke the glia of the retina, because classically in neurovascular coupling, we talk about a neurovascular coupling unit, and that involves 3 players: the neuron, the glia, and the vasculature. And so it seems in our imaging that we're skipping the inner retina and going straight from the photoreceptor through the Mueller cells or potentially also astrocytes to the vasculature. And there's been a lot of work on neurovascular coupling in the field, of course, as much in the brain as in the retina. A number of pathways have been identified that modulate both in terms of vasodilation as well as vasoconstriction. Parasites play an important role at the level of the capillary. And so I'll just remind you briefly about the nature of the circulatory system in the retina. We don't believe we are modulating the choroid. that we're looking at the retinal circulation as well as these capillaries embedded within the neural retina itself. Some imaging from in the literature showing the capillary bed in the retina. And now this is our very confusing drawing of the neurovascular coupling unit in the retina. Here we have the rod. It's transducing light. There are now multiple signals, not just glutamate. There are other signals that are released when the rod is active. And so there's this transmission of the signal from the rod into the Mueller cells, and then they affect the circulatory system here, the capillary, endothelial cell, a parasite, so forth. So there's a number of pathways that have been identified, and we haven't hit all of them, but we've hit a lot of them. All these yellow tags indicate pathways that we have hit with intravitreal injections of agents. And I'll just focus on 3 of them. First, glass, the transporter, the transmitter transporter, then also the nitric oxide pathways, and finally the arachidonic acid pathways. So first, glass. Oh my God. How do we shut this off? No. Okay, enough of that. First, glass is amino acid transporter, which one of the big jobs of the glial cells, of course, is to grab neurotransmitter from the environment, suck it in. And you see, unlike the neuroretinal agents, there's a huge impact on the imaging signal with an agent that blocks glass. It converts the signal into a vasoconstriction signal. Okay. I'll just move quickly with these. Nitric oxide, again, huge impact on the signal. And nitric oxide is known to be able to mediate both vasodilation as well as vasoconstriction. And it's a relatively slow time course. This is 15 minutes after injection, and you see a huge vasoconstriction response, and then later a return of the vasodilation response. And finally, the arachidonic acid pathway is very key. In its metabolites EAT and prostaglandins. So here's a prostaglandin blocker, indomethacin, and it abolishes the signal because EAT is, or the arachidonic acid metabolites mediate the modulation of vasodilation. So indomethacin is blocking the signal, and here it's returning after a time. And PPOH is blocking the creation of EAT, the other arachidonic acid metabolite, and again, it abolishing the signal here to a great extent, which returns over time. Here, 4 hours later from the initial injection. So what have we said? We've said that these pathways that have been identified as key in neurovascular coupling apparently play a big role in the control of our signal in the retina. And therefore, we believe that the neurovascular coupling pathways in the retina pretty much conform to that as described in the rest of the brain. And also will potentially serve as potential points for therapeutics. Okay, therapeutics. So why would we be interested in therapeutics? Of course, I told you I started off with thinking maybe this is good for glaucoma. Turns out probably not going to be. AMD is definitely a possibility, retinitis pigmentosa. But now we're thinking, in addition, instead of just worrying about the retinal function, maybe this method is also a great method for looking at neurovascular coupling itself in the CNS. And there's many CNS and other disorders where neurovascular coupling is a key a key dysfunction early, Alzheimer's, Parkinson's, and multiple sclerosis, PI. These all have early neurovascular coupling deficits. And if we had a good, inexpensive, non-invasive way of looking at the state of neurovascular coupling early on, it potentially would have a great clinical impact. But we start with diabetes, which is known, of course, to be substantially a microvascular disease. And, you know, people can end up with diabetic retinopathy 10 years after the original diagnosis of diabetes. It takes a while, but it's been implicated that a very early stage of diabetic retinopathy is a defect in the neurovascular coupling. Here in this review, they discuss methods for assessment, and part of what I'm going to be saying here is perhaps we should test out and add yet another method of assessment that's probably going to be more sensitive than, for instance, multifocal ERG, because it looks at neurovascular coupling itself directly. All right, so all these descriptions in the literature of what are the early mechanisms for diabetic retinopathy, they all involve early microvascular impairment and the breakdown of the hemodynamic response. Basically, what we're saying is if the neurons can no longer recruit and control its own blood supply, then they get sick. In an early stage, it's known as parasite loss. But what comes before what is, of course, one of the key outstanding questions. Here we see in a human with non-proliferative diabetic retinopathy, some dysfunctional degenerate capillaries, as well as ghost parasites. And already in the literature, the notion that vasodilation is dysfunctional in diabetic patients. Okay, so for this last vignette, we turn towards the rat because of a very well-developed model for diabetes using STZ in the rat. Here is then showing that our method for retinal imaging works very well in the rat. A large signal that's bigger than in mice. Part of the reason I'd like to use rats rather than mice. The signal is larger. And yet pretty much the same has been demonstrated in our other species. And now here is what happens when we look at our functional imaging neurovascular coupling signal in a diabetic rat. So before STZ-induced diabetes, we see a nice robust signal that you're probably sick of now. You've seen it many times, this slow development of a couple seconds functional response of the retina to a visible stimulus. Now, within a week or so of hyperglycemia, about I think 300 to 400 micrograms per deciliter, you see a dramatic impairment of this functional signal in the retina. And then out 3 weeks after STZ, the signal is basically gone. So this is highly suggestive that our method of non-invasive imaging of neurovascular coupling in the retina should be useful to detect the state of neurovascular coupling that would be relevant to diabetes, diabetic retinopathy, and we now also want to move to things like Alzheimer's as a possible platform for finding utility of this method. Okay, this is more STZ data. You see out there in 3 weeks, the signal's just way down. All right. Well, what have I told you today? I've told you that there's this relatively novel method for retinal imaging that's an outgrowth of the work we did in visual cortex. It is also a negative reflectance change. It's a small signal, Now we believe it's driven by outer retinal activity, specifically the rods. We know that it's we're really looking at a hemodynamic process. That is, we're tapping into neurovascular coupling. It's present in all the species we've looked at, notably humans. It's non-invasive and inexpensive. Pharmacological agents that impact neurovascular coupling impact our signal. And as a first stab at looking at early neurovascular coupling deficit in disease, diabetes, it does seem to have the expected result. And so potentially could be a useful tool for these kinds of diseases where there's early involvement of neurovascular coupling. So, all right, just wanted to thank you for listening and acknowledge a number of collaborators and key participants in this body of work, people from my lab, people in University of Iowa, and other contributors around the country. So thank you again, and happy to take any questions.
Good morning, Dr. So. This is Andrew Brown from UC Irvine. I really enjoyed that talk. That was really fascinating. I was wondering if you could expand a little bit more upon why you think it's definitely not the choroidal involvement in the signal. You could certainly look back at these different animal models and see the thickness of the choroid, or even, you know, if the choroid is supplying the RPE and the photoreceptors their main energy and oxygen source, surely that would be a larger contributor. And the second question is relating to your use of, I can't remember if it's nimodipine or nifedipine, the calcium channel. Nifedipine.
Yeah.
So it was really interesting to see that the time course of effect on your signal was in the course of about an hour, whereas the mean arterial pressure was adjusted for a much longer duration. So it seems like whatever the neurovascular adaptation in the retina that is affected is able to overcome that systemic administration. Quickly than the system is able to.
That may be the case. Yeah, I don't have too many comments about the nifedipine. We can take a look at, but let me take your first question, the choroid. Yeah, well, the thing is about the choroid is that, yes, it supplies the majority of vascular resources to the outer retina, but there still is a appreciable fraction that is supplied by the retinal circulation. I think Linsenmeyer demonstrated something like 10 to 16% of the oxygen delivery that the outer retina uses comes from the retinal circulation, not the choroidal circulation. We do have the ability to image the choroid separately. It's enough of a distance back in terms of depth of focus in the optics that we can image the choroid separately, and we don't see any modulation there. I think classically, the circulatory system in the choroid is so different than that of the retina. I think people already had decided that the choroidal circulation is not modulatable. It doesn't respond to neurovascular coupling. There are several studies that have directly measured this. Tim Duong using fMRI of retina, if you can believe you can do fMRI of such a thin tissue, but he's done it. And he's shown that the bold signal, at least in fMRI, is due to the retinal circulation, not the choroidal circulation. There are other methods for looking at circulatory behavior in the retina. One is laser spectral contrast imaging, and that has also, with the help of collaborator Randy Carden at University of Iowa, shown that it really is the retinal circulation that has the most of this functional modulation, not the choroid. So at least, you can never say 100%, but our definite bias is towards, or let's say it's away from the choroid as being the source of any of these signals. As far as the nifedipine question, yeah, I think what you're saying is that it appears that there may be some restoration of the intrinsic signal even when mean arterial pressure continues to be low. If that's what if I understand your question correctly. I can't really comment too much about it, but yes, maybe there is a suggestion. There is some rebound effect or some ability of the neurovascular coupling mechanisms to recover some of their function, even in the face of systemic low mean arterial pressure. So yeah.
All right. There is a question from Steve Lister. Would you like to take it over, Steve?
Sure. Thank you, Christina. Dan, Steve Liesler in Buffalo,
Hi, Steve.
really enjoyed this talk.
Yeah.
I had a question. At the outset, you talked about the work with the cat, and
Yes.
I was just wondering if there's any contribution to the response from the tapetum, the reflection.
Right. So I've looked at this question. The literature is scant. I can't profess to be an expert on it, but it's my understanding that the tapetum is non-reflective to near-infrared light. But it should be reflective, of course, to our visual stimulus. So that should give us about a factor, at best, a factor of 2 improvement, from the light rebound, the second pass, so to speak, of the tapetum that would help the cat response be larger. But is that partially what you're asking?
Yes, that was exactly where I was going.
Right. So yes, I think that could well be some of the explanation for the larger signal in the cat, the second chance notion of the tapetum. But as you know, the cats have better luminance efficiency than humans, that cannot be explained just by the tapetum. Cats are somewhere between 4 and 6 times better at seeing in the dark than humans, and so only a part of that is the tapetum. Others must be, somewhere in the transduction gain of the cat.
Good. Thank you.
Yeah.
Krishna, do you have a second?
Yes. So there is another question. Dr. So there was lately, this is Grażyna, and I'm here at UCI.
Yeah.
And there was a lot of publication lately about also the intrinsic optical signals, but originating from the elongation of the
right.
to the outer segment.
Yes.
Could you comment on that?
Well, there are a lot of intrinsic optical signals, a lot of sources of optical changes, light scattering, birefringence, that are related to function. So yes, I've learned about optical signals due to changes in the length of photoreceptors. There's early on, you can in this optical imaging work, Amiram experimented with in vivo measurements, which means there's no blood, and yet you can still see in the near infrared lots of functionally related signals, which he attributed to changes in membrane conformation in the movement of water. It's been known for a long time. I think David Kleinfeld has worked on this, where you can just see these very fast signals in neural tissue that are that follow action potential propagation. So all in all, what I'm saying is that there's many sources of intrinsic optical signals in neural tissue, and we're just describing one that we believe is very closely tied to neurovascular coupling, but we're aware of others. Another one that's been useful in the brain is flavoprotein autofluorescence. That is also another functional signal that can be picked up with optics. So yeah, I'm definitely aware that there's multiple additional optical signals out there to be measured with various properties.
Thank you.
Hi, Dr. Sal. Thank you for the talk. This is Francis Aguas. I'm a grad student here at UC Irvine. I had a question about the positive change in intrinsic signal that you touched upon. I'm curious if this response was consistent with increasing stimulus duration and the location of the stimulus. Did it similarly change the way that the negative signal changed? Thank you.
Yes, this positive signal still mystifies us a bit because it's always it's asymmetric. It's not on both sides of the original darkening, but it's always asymmetric. And so we did experiments trying to position the stimulus around various parts of the retina to see if it follows the path of circulation. And it does seem to be related. That is to say that the positive signals always seems to be more proximal to the originating source of the circulation, such as the optic disc. So our best explanation at the moment is that it's some form of vascular stealing. It's an effect that has MRI as well. It does not seem to be present in primates or rodents. So I can't really answer too much more than that, other than the things that you mentioned, such as increasing in signal size with increasing duration and other signals, stimulus strength. Now, it also, in appearance, looks very close to the kinds of signals that we saw with our various functional manipulations, such as this here. So, yeah, I mean, this we clearly believe is related to a vasoconstriction, very striking. But we haven't, I guess the short answer is we haven't fully understood the entire nature of the positive signal, other than it's only in cats so far. Yeah.
Hi, Doctor. So this is Philip Kaiser. Thanks so much for the great talk. My question is related to Dr. Brown's question earlier. So it seems, given given sort of the way that the retina is perfused, that the signal, this neurovascular coupling from the photoreceptors to the myorglia, affecting the interretinal vasculature could be just like a signal to prepare the inner retina to function more whenever there's light coming in. Okay, if that makes sense. So my question is, have you been able to, if that's the case, if you would disrupt that, you may have a case where the inner retina maybe becomes fatigued, so to say, after, extensive exposure to light. So I was wondering if you had been able, or has anybody done any experiments, say, with treatment of endomethacin? Does the bipolar cell or the retinal ganglion cells kind of run out of energy faster because they're not getting as much blood supply?
That's all you said sounds great. Give yourself a I don't know, a brownie point or something. Yeah, absolutely. All the things that you mentioned were right along the lines of my thinking. And I've sort of skirted around one problem which arises because of for which what you said is what I think is an answer. So the thing with neurovascular coupling in the brain is that it's been described as being related to the release of glutamate. More neurons in the brain release more glutamate, astrocytes their calcium waves, and induces vasodilation. Yeah, that's the way it's in the brain. The problem, which I sort of neglected to mention in the retina, is that, yes, the retina is opposite. The retina is backwards. That is, in the dark, there's this tremendous release of glutamate, and it's only with light that the glutamate is shut off. So it's exactly the opposite. So how do you deal with a neurovascular mechanism that is designed for increases for glutamate when the retina is exactly the opposite. And so the conclusion, I mean, this is all just speculation, that I came up with is along the lines of what you said. That is, it could be that it's designed to be anticipatory, that the photoreceptor, that the mechanisms know that when the photoreceptors are active, that the inner retina is going to be called upon to process visual information. And so, yes, what you're suggesting, that perhaps a long-term treatment with indomethacin might also make the inner retina sick. I haven't done those experiments. I have to think about exactly how you do it. I mean, it's sobering that it takes 10 years for diabetic retinopathy to really show its overt clinical signs after the initial diagnosis of hyperglycemia. So, I guess the retina is robust in many ways. So I'd have to think about exactly how you test that hypothesis, but what you suggest is, in fact, right along the lines that I've been thinking of.
Hi, Dr. So my name is Emma Lissier, and I'm a postdoctoral fellow here at UCI with Dr. Timothy Kern lab. So my interest is on diabetic retinopathy. The question that I have is that it looks like the signal that you're getting from the vasculature, you saw that is coming from the blood flow, mainly hemoglobin. And the models that you're testing are chronic models of inflammation, like diabetic retinopathy and Alzheimer's. So have you seen any change on the signal if you have an acute model of inflammation where you have a lot of immune cells coming through the vessels?
Well, I guess the short answer is no. We haven't done anything that's super acute, but I mean, STZ has an effect on the order of days, a few days. So I don't know, it depends on what you define as acute, but we're seeing these changes as early as we look for them in the STZ model. We don't look any earlier than a few days, but that's when hyperglycemia takes effect, and immediately there's an impact on their neurovascular coupling. But going even shorter than that, do you have some ideas about what you might want to do?
Like LPS injection, to stimulate like a storm of neutrophils coming to the retinal vasculature that might change the signal.
And then what's the time course of that?
It's 2 hours.
2 hours. Okay.
For IV injection of LPS.
Did you say IV?
For the tailbane, yes, tailbane
Oh, I
injection.
see. So it's a
LPS.
systemic injection.
Yes.
And it's as fast as 2 hours you see an impact in the retina?
Yes.
Okay.
All right, we need to speed up. Last two questions from the room and one from the Zoom. So let's start with the Zoom.
Hey, Jesse, this is your question.
Oh, hi.
It's good seeing you, and it was nice hearing the history of intrinsic signal imaging. So my question was, you talked at the beginning about how this is an accessible technology now.
Yeah.
Given that CCDs and CMOS cameras are getting pretty cheap and pretty sensitive, what does it take for someone to get a fundus camera and a detector and doing similar work to what you've been doing? Is this scalable in the commercial domain?
Well, I certainly think it is. I think you could probably build an instrument for less than a few $1,000s commercially, and there's always a factor 3 built in there. That's what Amiram had always said, is whatever it's going to cost, multiply by a factor 3 for commercialization. So I mean, I think that clearly the answer is yes. I mean, you have these iPhone ophthalmoscopes and whatnot. So yeah. So the question is really how to get a stable protocol for human use. It's still been tricky because this is such a small signal. It's been tricky to get a nice, robust image from a human. So that's I think the toughest piece that we have to tackle before, real commercialization. But in terms of the technology, absolutely, I don't see any significant barriers. Yeah.
Thanks.
Yeah.
Hello, Dr. So this is Jan Skersetal.
Yeah.
I was wondering about the idea of decoupling the rods from the blood vessel network. And have you looked at clinical populations with rod dystrophies? And relatedly, have you thought about behavioral tests to capture differences across the field in intensity of the signals using rod isolated stimuli in humans, obviously?
If I understand your question, because of the non-uniform distribution of rods, you want to map out differences in signal that are due, if indeed it's a rod signal, that it should follow the distribution of the rods. Is that where you're going with that?
Well, if this is an early marker of change, then it should be captured as well in behavioral tests. That's my second question. And the first one is more proof of concept that in a rod dystrophy,
yes,
according to your model, that effect should vanish.
Go away.
I
Yeah.
understand, right?
Right. I mean, or you might be able to reveal a cone signal, perhaps that was otherwise buried because of the rods. So yeah, no, I mean, the answer is that we haven't pursued any of those possibilities, but they sound worth doing. Yeah.
Thank you.
Yeah.
Hi, Dr. Cho. This is Vladimir Kefolov. I'm in the audience here at UC Irvine. Thank you. That was an amazing presentation. And I had probably a dozen questions that popped in my mind. And then you answered pretty much all of them as you went along. But I missed maybe what you think is the signaling that couples the rod to the Mueller cell. So you showed beautifully that you need the rod phototransduction, and you showed that the Mueller glia are involved. But I missed the nature of the signal that couples, you think, the rod cells to the Mueller cells as
Yeah.
activation.
Very perceptive. Right. So that's because I still don't have specificity about that. I did mention that there's a number of things other than glutamate that are released when photoreceptors are signaling. And we suspect that it may be one of those agents, but the actual identity of the coupling between photoreceptor and glial response, we still don't have a clear handle on it. Some people have suggested, calcium shifts in potassium concentration. That's a possibility. There's other agents such as taurine that get released. But no certainty yet. Definitely one of the outstanding questions out there.
Thank you.
Thank you, Dr. Tso, for the wonderful presentation. And we look forward to the next week, where we hope to see you all. Thank you.
Thank you. Thanks for the invitation.